Thanks everyone for being here. I would like to start by thanking the organizers and the steering committee. It's such a great opportunity to attend this conference and also to present my work. So I will be presenting you the work I did during my PhD, which I did at the University of Manchester and the Institute of Systems Biology in Seattle. And I will focus 2/3 of my talk on point 2, that is about non-Darwinian dynamics of drug resistance. So apologies, that's about 90%, 95% experimental work, so I hope you bear with me and don't get too bored. And then 1/3 of the last third of the talk will be about non-equilibrium population dynamics of phenotype conversions of cancer cells. So therapy resistance and tumor relapse after drug therapy are commonly explained by Darwinian selection of pre-existing drug-resistant, often stem-like cancer cells. And it's assumed that they result from random mutations. But what happens actually is that we have a huge non-genetic heterogeneity and plasticity of the tumor cell phenotypes. So we ask the question whether mutations are really necessary and sufficient to promote cell phenotype changes during tumor progression. So in the 50s, Waddington proposed that development occurs similarly to a ball rolling down a sloping landscape, which contains multiple hills and valleys. And the cells take different paths down this landscape, adopting different paths. But however, differentiation is not terminal. And the different cell states are maintained by epigenetic barriers that can be overcome given sufficient perturbation. So the most recent example of this is the induced pluripotent stem cells. So if we assume that there is a unique map between the genome and the gene regulatory network, then we can map this gene regulatory network into a state space and transform the metaphor of the epigenetic landscape into something that we can actually have a model on. So this gene regulatory network for organism A can be on state 1. So it will correspond to an attractor on the state space. But the same gene regulatory network change its expression values. So then it will occupy a second attractor of the state space. In normal development, what we have is, so the cells start at the top of the epigenetic landscape where they have higher potential, and then they go down and, well, they cross the progenitor cells, and then the terminal differentiated. But the gene regulatory network is multidimensional. So we have attractors that are normally occupied during development. So those are the ones that correspond to the normal cell types that the organism have. But because of the multidimensionality, we always have attractors that are there and are not occupied. So that's our assumption. These unused attractors are not shaped by evolution. So that's the difference, because on the normal attractors that the cells normally go, evolution has shaped them, so they have a proper regulation to have a normal behavior. These unused attractors, they are not well regulated. So if something happens that a cell can enter one of these attractors, then the phenotype is no longer predictable, and that is what we classify as a cancer attractor. So I will now jump into the non-Darwinian dynamics in therapy-induced cancer drug resistance. So as we have been listening through the entire week, the problem of cancer drug resistance is a old one. So we are all familiar with this picture. Then we have a big tumor to start. Then we treat, it shrinks, and then eventually it comes back. The problem is when it comes back, it usually comes back in a much more malignant form. So why does that happen? So far, we have been mainly explaining drug-resistant state evolution using a Darwinian dynamics. So we assume that we have cell 1 that has a genome 1. Then we need to call in a mutation to get genome 2. And that, if you see in here, genome 1 maps into this gene regulatory network. Genome 2 maps into the second regulatory network, and each regulatory network then maps into its own expression state, and the landscape has to change because there is a unique correspondence between the underlying gene regulatory network and the epigenetic landscape. However, if you consider a non-Darwinian dynamics, then you assume that you can transit from state 1 to state 2 without changing your gene regulatory network. So basically, you just change the expression patterns. Then you don't need to change the epigenetic landscape, and you can just change the state where you are. So having this unique mapping between the genome and the gene regulatory network, we can then propose that the drug is actually a perturbation that is able to induce a state transition. So if within the tumor we have sensitive cells and perhaps some resistant cells already in there, what we might be doing when we treat is actually shifting the balance. So we are inviting some of the sensitive cells to join the resistant ones and making the tumor worse. So our hypothesis is that because therapy does not kill all the cells, we perturbed these ones, and these switch to a more resilient state, and so we have the phenomena of therapy-induced drug resistance. So we were trying to kind of focus on multi-drug resistance because then we don't need to worry so much about the mechanisms of the drugs. And there are two ways to measure multi-drug resistance. One is with a direct measurement. So this is by an antibody staining, and I was focusing on MDR1. MDR1 is a member of the ABC transporters family, so it has been mentioned early in the week, is commonly known by P-glycoprotein. And the hypothesis is a sensitive cell does not express the protein, but after treatment, we will see some expression. So if we plot these in a y-axis, what we have is a sensitive cell here and a resistant cell there. But we also have a functional assay that works as an indirect measurement. So if we add the cells, something like a surrogate reporter, so it's a dye that behaves in the same way that drug does. So a sensitive cell is going to accumulate a drug, and then after treatment, this drug is going to be pumped out, so will the reporter dye. So we can add an x-axis, and then we have a two-dimensional way to look at the sensitive cell that takes all the dye and doesn't express P-glycoprotein, and the resistant cell that has high levels of P-glycoprotein, so it does not uptake dye. And I was working with AGL60 leukemia cell line. So this is a model cell line for AML. And when we have this protein, and this is a flow cytometry profile. Is everyone familiar with the flow cytometry profile, or should I explain? OK, I can just 30 seconds. So basically, on the x-axis, you have the expression of the antibody measuring, and this is a histogram, so histograms, you know it very well. Basically, what we can see in here is that it looks like a unique population, but you have kind of a second one around here. So what I did was I sorted the fractions of the populations, and then I put them back into culture. And surprisingly, the cells that come from this fraction, so the left one that corresponds to about 98% of your population, repopulate the entire population within 12 hours. And it's very interesting because then you can go back here, sort again, and you observe the same dynamics. But if you look at this right fraction here, after sorting, they go through a bimodal behavior, and eventually, after 17 days, they are able to reestablish the initial population. So is it just a differential growth? Not actually, because if you look in the presence of drug, what you see is that the cells that come from the right fraction have a much higher survival than the ones that come from the left fraction. So we actually have some resistant cells already in our population. So we were looking at whether are we just selecting them, because in drug they're going to have advantage growth, or are we actually allowing transition from the blue to the red? OK, so in order to do that, I did a time course of the cells, and I treated them with vincristine for this amount of days. And as you can see, both for the functional assay and for the MDR1 expression, after about 24 to 48 hours, we see a strong emerging on the population. So we have a higher percentage of the population starting to pump out the drug, and you see the same. So we have a fraction of the population that is now starting to express MDR1. So the chemotherapy is inducing the expression of the protein and the phenotype, and we could also measure that when we look at the RNA levels. So the plot on the left shows you levels of nascent RNA. So if you actually try to measure the RNA that is just being transcribed, it's just a strategy of how you design your primers, you can see if you're actually inducing or if you are, for example, just stabilizing your mature RNA that you might have already. But what we were able to show is that we have a massive increase within 30 minutes and 4 hours of the nascent RNA. And that trend keeps for about 11 days always going up, even if the drug has been washed away. So we just treat 1 day, then we wash, but there is kind of a memory effect in the system, and the system starts to relax at 17 days. So we went back to the drawing board, and we were trying to understand what is happening within the cancer attractor, because if this is a cell line for cancer, so they are all in here, but we have some drug sensitive and some drug resistant. So we decided to do a state transition model. So I usually work with biologists, so I have some cartoons around the equations to amuse them. And so this is a very simple ODE model. But the interesting thing in here is that we have these terms for growth, and the Ks are for transitions. But experimentally, we can determine the growth in either the untreated and the treated case. So by doing the similar experiment of sorting and then just measuring growth, I can do that without any treatment, and I can do that after treatment. So I can get 4 of the parameters that I need, but I cannot measure experimentally the transition rates. However, I can simulate the ratios. So this is what is shown in here. So I know all the ratios of the growth, and I can simulate all the ratios of the transitions. And I know that within 24 hours, the population has shifted to a 60-40. So I'm looking around on the color map for green. So I will be in here, and I know that the growth ratios are around 0.65, because that's what changes when we have control and when we have drug. So in control, the left side is growing faster than the right, but in the presence of the drug, we see an inversion like we would expect, because the ones that underline with blue will die more than the ones in red. So I know that I need this region here, and this automatically predicts that I need to have a transition. So I need to have a higher number of cells going from the low resistant to the high resistant, and not the high to the low. So this suggests that transition is playing a very important role. But this is all at population level. So the next question is to ask whether what we are observing is actually a cell autonomous phenomena. So the way to do that is you can load the cells with the reporter dye, and then you can ask the question if after treatment they're gonna release the reporter dye. So I did live cell imaging, and this one takes about 3 days, so the time is in there and the scale in here. At the beginning, the cells are all green, and then in about 24 hours, the green is going away. But the blue is the nucleus, so you're looking for this kind of pattern, is a healthy, happy cell. When they do this kind of blue condensation, that means the nucleus is kind of condensing and is about to die. And you can see that in like 65 hours, there is almost no green, although they start to regain some, which is interesting because that means the reporter dye is still active and they have reached the balance. So there is some going in, but I will just kind of quickly play this again. At the beginning, it's much, much brighter. So basically, the reporter dye is still there. It goes in but goes out at like it kind of reach a steady state, which is very interesting. So we have identified that we have different subpopulations. So we did microarrays of untreated and treated case. And surprisingly, when we do the self-organizing maps for the entire transcriptome, they are not actually very different. So you have some differences at the level of the immune and the inflammatory response, some differentiation inhibition, but globally, they look very much the same. However, if we dig in into the treated population, we fractionate and we do microarrays of the subpopulations, then we see that this, that is the side of the population that is induced after drug, is massively different from the one that is kind of aligned with the untreated population. And in here, we have something very interesting, like the Wnt pathway, polycomb, and some cytokines, BRD4. So this is a kind of a strong stem signature that we are getting on. So we decided to then focus on the Wnt pathway that is highlighted in here. So these were all the candidates that we got from our microarray data, and we decided to knock down beta-catenin because beta-catenin is the central element. So beta-catenin is what actually, when the Wnt pathway is active, is what actually goes into the nucleus and binds the TCF group to start the transcription of the target genes. So if we knock down beta-catenin, we actually observe a reduction on the induction of the phenotype, both at the functional level and the MDR1 expression as well. So we can conclude that the drug-induced resistance and the MDR1 expression correlate with the upregulation of the Wnt signaling pathway. The next question was, if this is not a genetic phenotype change and we're actually looking at transition, is the phenotype reversible? So what we did was, like, I treated the cells for one day. They were actually like in drug for 2 days here, and then I washed the drug away, and then I followed the relaxation. And like I have seen before, they take about 17 days on the global picture to come back. But doing microarray analysis at all these time points, what we observe is that there is a memory effect that is probably easing off about 17 days, but the population still remembers all the states that you went in through, even if the drug has been washed away. So the question is, how does the system remember? So again, I treated for 2 days, then I washed, and then I retreated. And what is interesting is, given for a certain dose of pre-treatment, and then the survival on the re-challenging is proportional. So I re-challenge them with 10 nanomoles of drug, that is this here. So the ones that I have pre-treated with 10 nanomoles, almost all of them survive. And the control, only 20% survive after 3 days in drug. So the drug-induced resistance is non-genetically inherited across cell generations, independently of the presence of the drug, at least for a limited period of time. So for this part, the concluding remarks I would like to do is that the analysis of the MDR1 induced phenotype provide evidence of drug resistance that can be induced by a non-Darwinian selection principle. And I've shown it experimentally with single-cell longitudinal monitoring, demonstrating cell individual adaptation. I've decoupled the expression from the function to exclude possible Darwinian selection. And we have the transcriptome analysis revealing a sustained genome-wide stress-induced state. So the question now is, that was all done within 1 to 3 days and then just looking at it, but what if we adapt the cells to drug? So in here, in orange, is cells that have never seen drug, and in red is cells that have been in drug for almost 1 year. And after that, what I do is I wash the drug away, and I follow their relaxation. And what is very interesting is that in about 100 days, the phenotype is almost all back to the initial state. So the blue line, that is the ones that were red and then I washed the drug away, is almost all overlapping with orange. So this is, we can show that even the long-term cultures can relax, but then is there something else aiding to this phenotype? So I then look into a different side of drug resistance, because most of multi-drug resistance is looking into efflux. So what if you look into influx? How is the endocytosis of the cells actually behaving? And what I was able to show is that the fluid phase endocytosis, so the just normal way of, just like without any mechanism or without any carriers, just like our normal liquids diffuse into the cells, is actually smaller as well in the long-term drug-resistant cells. So basically, if we block endocytosis by putting it at a lower temperature, we observe the same phenomena. So actually, the resistant cells have also downregulated their endocytosis to prevent drug accumulation. So we can see that very nicely at single-cell resolution. So on the left, I have the parental cells, and on the right, I have the drug-resistant cells. And you see that here, the drug is accumulating, while on here, it is not. So if we quantify that, we see that there is a significant difference between the two lines. And then I went on and asked if there was a general fluid phase endocytosis block by loading the cells with phytC dextran, that is what is commonly used in the literature. And yes, there is a general fluid phase endocytosis block. So the uptake is much reduced, and a drug that can just stimulate endocytosis by just kind of permeabilizing the membrane is able to recover the phenotype. And this was all done on the reported dyes to show the principle. But what about the chemotherapeutic drugs? So in the chemotherapeutic drugs, we observe exactly the same thing. So if we treat the cell lines with doxorubicin accumulates on the controls, but is not able to accumulate on the resistant cells. And although we also have a contribution of the efflux, we can very confidently say that a big shift in here is mostly due to the differences on the uptake. So with this, I uncovered a new facet for drug resistance. So the fluid phase endocytosis is directly linked to long-term drug resistance, and the ABC transporters have two protective roles for the cell. So they can efflux, and they also can trigger an adaptation to prevent endocytosis. So the take-home message from this part of the talk is that therapy never kills all the cells, but the non-killed cells are actually perturbed. So they switch to a more resilient state, and these stressed dying cells can release signals that can stimulate further state transitions. And so the multi-drug resistance is epigenetically inherited across generations. By doing this, we're actually opening space for some selection and for some phenotype that can become established through mutation, perhaps. And then the phenotype is reversible. So a corollary from this is different anti-cancer therapy schemes, like we have been listening through this week, might actually lead to weaker surviving cells, because with our data at the moment, what we're showing is that if we have an early growing tumor that we treat, we actually make it worse. But then if we could have something like a stress response blocking therapy, then perhaps we could recover it. But we might be able to avoid this relapsing tumor if we just treat it as a chronic disease. So now I would like to change gears a bit. And before I start, I will acknowledge Joseph. So the work that I will present from now was done in a strong collaboration between me and Joseph. There is a postdoc at my PhD lab. And so this is a modeling work. So please don't ask very hard questions, or please feel free to do. So this work is a non-equilibrium population dynamics of phenotype conversion of cancer cells. So the current mathematical theory of cell population dynamics is mostly developed based on the Luria-Delbruck theory. But there, the population heterogeneity is actually due to the diversity of the genotypes. So they always need to have mutations instead of multistability to actually have their different cells. So they actually don't acknowledge non-genetic transition between multiple attractor states. So they assume a fixed one-to-one mapping between the genotype and the phenotype. And actually, that's not always the case, because the entire cells of the body have the same genotype, but their function is completely different. So that assumption is at some point questionable. So with recent studies on mammalian cell biology, if there is more and more evidences that multistability has a strong role. So on this model, we have departed a bit from the theory by implying that, that we need to have reversible state transitions. So the transitions are often symmetrical, and the mutations are, well, the mutations are typically irreversible. The state transitions are frequent, so the rate is often at the same time scale as division, while the mutations are much slower. And the transitions are not strictly non-Lamarckian. So we actually allow induced transitions, so something like you know where you want to go, like if the system is being perturbed in a conscious way rather than just the stochastic selection by the Darwinian mechanisms. So we went back to our favorite two phenotypes that you might have realized by now, because it's very simple to kind of start working. So we include the growth terms explicitly, and this is the big novelty of the model. So having both growth and state transition enables us to then look at the different subpopulations of the main cancer population. So we just wrote a simple ODE, matrix form, and then general solution. And then we looked at how the long-term behavior is, and that is given by the eigenvalues. So if the eigenvalues are both negative, then the growth term associated with the negative eigenvalue decays exponentially. So the two cell populations have essentially the same growth rate, but only one subpopulation can survive independently, while the second subpopulation lives as a derivative of the other one. But if the product of the eigenvalues is positive, so we can have both eigenvalues positive, and the two subpopulations survive together. And in the long term, they grow with the same rate, which is defined by the larger eigenvalue. Or if they are both negative, both subpopulations are going to become extinct together. So mathematically, the product of the two eigenvalues is the determinant of the matrix T. The condition for the two eigenvalues having the same signal is given by this equation. So we have two possible scenarios. We have the scenario that the growth rates for both phenotypes are much greater than the state conversion, and so the two subpopulations can both survive on their own. However, if one of the two populations has a transition rate larger than its division rate, the population can only survive as a derivative of the other, because it depends on the backflow. And the distinction of the two discrete subpopulations is becoming blurred. So we can actually consider these two populations as a single one that have a mean growth rate. And this is the interesting point, because when we have these, then we enter the domain where the non-genetic phenotype conversions actually dominate, because it's all about transition from one state to the other. So, and when we can calculate the population ratio, and then while the population ratio becomes stationary, both populations can actually grow indefinitely. So this result deviates from the classical population dynamics, because the coexistence of the two populations of different growth rates is not stable due to one direction conversion mutation in the classical theory, but actually here they can both grow and the population is stable. So the changes in the growth rate relative to each other are actually representing differential fitness in the given environment, which can promote a Darwinian selection, but the changes in the transition rates can represent a Lamarckian induction, in the sense that a given environment might impose differential transition rates between different phenotypes. So this is what I was mentioning about the implications. And the interesting thing about this framework is that it can describe the relative contributions of both Darwinian and Lamarckian during tumor progression under chemotherapy. But what happens is, normally during development, we have multicellular organisms, so we form usually more than 2 cell types. So we have M phenotypes, and we can generalize our equations, again do a matrix form and get a general solution. But in here, there is no non-trivial steady state. So this becomes very interesting, because the coexistence or the coextinction of the M distinct cell subpopulations requires that the M eigenvalues that are either all positive or all negative. So we need, in order to maintain the distinct cell populations, we need the growth rates to be much larger than the transition rates. But if one of the M populations that we kind of described in here, if one of them has a bigger transition rate, then it's all gonna again depend on the backflow from the others. So the consequences from here is that the influence of the growth term in the cell population dynamics is that the dynamics of a state X are captured by both transition rates and growth rates that vary from state to state. The growth rate is not included in when the growth rate is not included in the reequilibrium, then we guarantee that we have we always have a steady state independent of the population ratios. But when the growth rate is considered, there is no unconditional reequilibrium for any transition rate. So to maintain 2 distinct cell populations, transit-onto rates have to be much smaller than the growth rates. And so the implications for selection and instruction is that a macroscopic change of the population property X in a particular environment in one direction can in principle be achieved by selection. So in the presence of S, the cells that happen to be in state Y have a higher growth rate, and there is no actual change of gene expression in any cell. Or by instruction, so a state S induces a gene expression pattern change in individual cells that confers a new phenotype. So this is an individual cell level event, and is a change of the molecular network state of a cell. So in evolution, this is kind of the Darwinian Lamarckism dualism. So to wrap up, by uniting the dynamics of reversible transitions between distinct cell phenotypes with their growth rates, we can capture the contributions for the evolution of the cell population from both Darwinian selection and Lamarckian induction. We have a mathematical framework showing that both growth and transition influence the ratio of cancer cell populations, and we derived the condition that different cancer cell types can maintain distinctive subpopulations. And while Lamarckism has a little role, if any, to play in organism evolution, in our model, we have the first attempt to demonstrate that, in principle, Lamarckian and Darwinism are 2 different sides of the same coin. And this not only applies to the study of cancer cell dynamics, but it can also help us understanding of the emergence of drug resistance in anti-cancer therapy. So I would like to finish by thanking my PhD supervisor, Professor Suyu Wang, Joseph, which I mentioned before, and all the people that I worked during those 4 years, and also now my current boss, Professor Fiona Watt, and my funding from King's College, and you all for listening. And if you have any questions, I'm happy to take them.